everyone, to the Chemistry of Life Processes Institute first Chalk Talk of the year with Professor Michael Jewett. For those of you who are unfamiliar with the Chemistry of Life Processes Institute, CLP is a nexus for interdisciplinary biomedical research, innovation, and training. More than 65 faculty members from across Northwestern belong to the Institute and collaborate across disciplines to develop new therapeutics, diagnostics, and other technologies. And we provide interdisciplinary training for the next generation of researchers through our NIH training program and undergraduate research program. It's my pleasure today to introduce Professor Michael Jewett as our speaker. Mike is the Charles Deering McCormick Professor of Teaching Excellence at Northwestern University and the Walter P. Murphy Professor of Chemical and Biological Engineering. He's also the Director of the Center for Synthetic Biology and an Institute Fellow of Northwestern Argonne Institute for Science and Engineering. He has been a resident member of CLP since 2009, and his lab is located on the first floor of Silverman Hall. Mike received his PhD from Stanford University, completed his postdoc studies at the Center for Microbial Biotechnology in Denmark and the Harvard Medical School, and was a guest professor at the Swiss Federal Institute of Technology. He's the recipient of many honors, including the NIH Pathway to Independence Award and the David and Lucille Packard Fellowship in Science and Engineering, along with multiple Young Investigator Awards. As you will learn in today's talk, Mike's lab has been at the forefront of synthetic biology, and their innovations are impacting biomedical research, materials science, and energy research. So that's the end of my introduction. Before Mike gets started, I want to make note of a few housekeeping items. First, if you would please keep your microphones muted throughout the presentation, that way we can minimize any unintended interference. Please feel free to turn your video on during the Q&A period. After Mike's presentation, there'll be about 15 minutes for Q&A. Please use the chat feature at the bottom of the screen to submit your questions, and Lisa LaValle will be moderating. This program is being recorded, so we can share this with those who were unable to participate today. So now it's my pleasure to welcome Mike Jewett.
Thanks, Sheila, for that really kind introduction, and happy St. Patrick's Day to everybody. Hopefully you're doing well, and it's really my honor and great privilege to talk a little bit about the effort that our lab has been pursuing in bioengineering beyond cells to enable a fair and sustainable 21st biocentury. Before I get into things, I do have a few conflicts of interest, and so some of the activities that we pursue in the group can benefit from the research here. So taking a step back, I think that we as humanity are facing challenges of a magnitude that we've never seen, right? So human-caused climate change is altering the health of our planet. At 1.1 trillion tons, man-made materials now outweigh all living systems on Earth. 30% of the world's population lacks access to essential medicines, and more than 2 billion people lack access to clean water. And by combining disciplines and exploiting the intersections between them, synthetic biology has become one of the most promising fields of research to address these challenges in planet and societal health. Even most recently, President Biden, in a letter to the Office of Science and Technology Policy, listed synthetic biology along with artificial intelligence as two of the industries of the future that will lead to positive change for society. If synthetic biology is to make an impact, what should we build? What can we build with synthetic biology? How can we ensure that what we build facilitates a fair and sustainable biocentury for our planet? These are the types of questions that many in my group have been wondering about, and today I'd like to share a few vignettes focused on these types of activities about what we can build with biology today. And I'd like to start with a effort that's focused on therapeutics, something that connects closely to the Chemistry of Life Processes Institute, and specifically protein medicines. Protein medicines have revolutionized our ability to prevent and treat human disease. From the manufacture of antibodies to vaccines to hormones to enzyme replacement therapies, medicines have really transformed the lives of millions of people. Yet, the World Health Organization estimates that about 30% of the world's population lacks access to essential medicines. Why? One of the main reasons is that our current manufacturing paradigm limits the ability to distribute protein medicines. We oftentimes make antibodies or other protein therapeutics in large centralized facilities, such as this plant shown here, where we culture microorganisms or mammalian cells to facilitate the manufacture of these protein products, purify them, and then distribute them, typically with a cold chain, to the point of use. Our ability to produce and deliver medicines is therefore limited by the expense and time required to build such facilities, which can cost hundreds of millions to billions of dollars and take multiple years of time, as well as the feasibility of refrigerated supply chains. And we don't have to look any more closely than the recent COVID-19 vaccines to have an assessment and understanding of the challenges associated with this, right? As the first Pfizer and Moderna vaccines rolled out, there were questions about the shelf stability of these medicines. Even more recently, with the vaccine rolling out from Johnson & Johnson, there wasn't enough capacity to manufacture it. So we made partnerships with Merck so that Merck could help additionally provide manufacturing capacity of these centralized facilities. So we and others are getting interested in thinking about how can we provide low-cost alternative and portable biomanufacturing platforms that broaden access to medicines, that might rethink this centralized paradigm that we have and allow humankind to make medicines when and where they're needed. The way in which my lab pursues this effort is using something called cell-free systems. So cell-free systems enable portable, decentralized manufacturing. So how do they work? What we do is we take a cell, we rip off the cell wall, and we collect the insides of the cell. Basically, we centrifuge out the cellular wall components. And in so doing, we then feed the extract or the proteins that are left over with raw materials, amino acids, substrates, energy substrates, information. And that allows us to enable molecular transformations to synthesize protein therapeutics. You can kind of think of this like taking a car, lifting the hood up, pulling the engine out, and then repurposing the engine. What we're doing is repurposing the engine of the cell, the molecular machinery of the cell that makes proteins. And over the last couple of decades, we've seen continued increases in our ability to enable cost-effective, high-yielding protein expression from these cell-free systems, now allowing us to make proteins on the order of 2 to 3 grams per liter, which is commercially relevant. What's even more exciting is that these cell-free systems can be freeze-dried, just like you might buy freeze-dried yeast from the store to bake bread or to brew beer. We can freeze-dry these cell-free systems, store them, distribute them, and activate them by just adding water. So we can take these cell-free systems, create freeze-dried extracts, imagining ourselves to transport them to the point of need, just add water, and allow us to produce a variety of different products. My lab's been interested in thinking about these cell-free systems then as a portable biomanufacturing platform for the synthesis of vaccines. A particular model that we started with is something called a conjugate vaccine. Conjugate vaccines can prevent life-threatening bacterial infections, and the way in which they work is they train your immune system to recognize a pathogenic bacteria. On the outer wall surface of bacteria, there are sugars. These glycans or polysaccharides can be used as a fingerprint to teach your immune system how to recognize a pathogenic bacteria. What we do is we take those polysaccharide antigens and we attach them to an immunological carrier protein. And that creates a conjugate vaccine. It's simply this carrier protein attached to a polysaccharide antigen. These carrier proteins typically represent molecules that your body already recognizes as foreign. So for example, diphtheria toxin protein, tetanus toxin protein, proteins your body may have seen, for example, in a DTaP vaccine. What's exciting about conjugate vaccines is they're already known to be very effective and safe. And in fact, the approved conjugate vaccines, they typically demonstrate about more than 90% effectiveness. One of the challenges, however, with these conjugate vaccines is that the current manufacturing methods limit distribution of them because it's technically challenging and costly to culture pathogenic bacteria. So if you have pathogenic bacteria that could be harmful to humans, you have to grow them up in large reactors and then strip off the sugars so you can use those for conjugation. This just needs to be managed very efficiently. You do that, of course, in expensive, highly centralized facilities, and they do require cold chain distribution. We wondered, and in particular, my student Jessica Stark, who's now working as a postdoc in Carolyn Bertozzi's laboratory, along with our collaborators, Matt DeLisa and his student Tommy, shown here, wondered if we could create a freeze-dried, decentralized manufacturing platform for producing conjugate vaccines. This in vitro conjugate vaccine expression platform works like this. We take a cell and we would make it glycocompetent so that it would have all the necessary components for glycosylation. We'd freeze dry that, allow us to transport it to a location without refrigeration, add water, and use that to deliver a medicine. Now, if you were dreaming of what you would wish for in a decentralized manufacturing platform, you'd probably come up with a few key features. First, you'd want it to be fast. Hey, can I make a medicine in less than an hour? You might want it to be modular, so you could synthesize different carrier proteins, but also vaccines against different bacteria. You'd want it portable for cold chain independent distribution, safe for injection with minimal purification steps, and effective for producing vaccine products at scale. Now, I'm not going to show you each of these steps, but in a recent paper we published in Science Advances, we actually went through, and Jess and Tommy demonstrated the ability to innovate solutions to each of these features. We did this in the context of a model bacteria, Francisella tularensis, which is a highly infectious pathogen. About 25 colony-forming units can be lethal. And the idea was, let's take the biosynthetic pathway that makes the sugar fingerprint, that polysaccharide O antigen, and we'll put that into an E. coli cell, as well as a plasmid encoding an oligosaccharyl transferase. Now, if you want to make a glycoconjugate protein, you need three parts. You need, first of all, the protein you're going to decorate. You need the polysaccharide antigen, but you also need this enzyme I just mentioned, an oligosaccharyl transferase that attaches the polysaccharide antigen to the protein. So what we did is we enriched an E. coli cell with the pathways for these components. We created an extract of these cells. We grew the cells up, ripped off their cell walls, removed the cell walls, and we're left with the soluble components. That allowed you to then add a model plasmid to express a carrier protein and ultimately make a conjugate vaccine in less than an hour. So the gel on the right-hand side shows you by Western blot that we're able to make conjugate vaccines for Francisella tularensis. So I mentioned we needed three main parts, right? We need an oligosaccharyl transferase. In this case, we use an enzyme from Campylobacter jejuni. We need a polysaccharide O antigen here from Francisella tularensis. And we then need a protein that we can decorate, plasmid DNA encoding that protein and a sequence of amino acids that can be decorated. And this Western blot highlights two features. One, the red color is showing you the protein we decorated. In this case, it's just green fluorescent protein, everyone's favorite model protein. And the green lines in this banding pattern you start to see indicate the polysaccharide O antigen attached to that protein. And what you see is only in the instance where we have each of the components that are necessary, the oligosaccharyl transferase, the polysaccharide antigen, and the plasmid DNA encoding GFP, do we actually get conjugate protein production, where the glycoconjugate is attached to the protein. So this is exciting because it validated that we could make, in this case, a glycoconjugate candidate protein in our cell-free protein synthesis system. But we didn't know how robust it was. And so what Jess did is she actually explored a variety of conditions and physicochemical optimizations to demonstrate that we could actually produce these glycoconjugate candidates in our cell-free reaction at not only 37 degrees, but 25 degrees in room temperature. And in addition, we could do that fast in as little as 15, 30, or 60 minutes. So what you're seeing here in this Western blot is in the absence of the oligosaccharyl transferase, we don't make the glycoconjugate. And in the presence of the oligosaccharyl transferase at each of these times and temperatures, we actually are making the glycoconjugate. So the system is robust. It works at different temperatures. It's very fast. It works in less than an hour. It also could be used to make glycoconjugate proteins for, in particular, FDA-approved carrier proteins that I'm not showing. And it could also be portable. So we wanted to demonstrate that we could freeze dry this system for distribution. And so what we did is we took our cell-free protein synthesis reaction, we freeze dried it, so we removed the water from the system. And we asked the question, could we still make this glycoconjugate protein from a freeze-dried setting? So in this case, this gel is showing us that in fact that's the case. We have a fresh system, so a liquid-based solution system that was never freeze-dried, one that was lyophilized. And what you're again seeing is in the absence of the oligosaccharyl transferase, we don't see this ladder-like green banding pattern of the polysaccharide antigen. But in the presence of the oligosaccharyl transferase, we actually make a glycoconjugate, both in the fresh system and the lyophilized system. And importantly, in the lyophilized system, we get about 75% of the activity. And we found that the system was shelf stable for about 3 months at room temperature. So this was exciting because we could make not only proteins in less than an hour under robust conditions and demonstrate, although I didn't show you, expression of a variety of FDA carrier proteins, but we had the system that was portable. We also made this safe, so we addressed issues in endotoxin activity. And when we put all that together, we actually were able to deliver a manufacturing approach that allowed us to synthesize conjugate vaccines that were protective against this model pathogenic bacterial challenge. So what we did is in a mouse model, we produced conjugate vaccines using the protein D of influenza as our model FDA-approved carrier protein. We attached to that the O antigen, the polysaccharide O antigen from Francisella tularensis. We delivered that as a vaccine to a mouse with 2 boosters, and then challenged with a lethal dose of this bacterial pathogen, Francisella tularensis. The important point to take away is each of the mice that received our conjugate vaccine survived. And so we had a 100% survival rate in 2 different studies with this particular conjugate vaccine that we made. So where are we? So we started to be able to create this cell-free platform for portable, on-demand, and scalable production of conjugate vaccines. I mentioned we could freeze dry these extracts and they were shelf stable for about 3 months. And our protein yield was about 20 micrograms per milliliter. I'll come back to that in a second. What that means is in about a 0.5 milliliter reaction, we could make enough dose for a single person. Right now, it costs us about $5 to do so. And that actually compares favorably with other conjugate vaccines that are on the market. So for example, Prevnar or others, where typically these prices are between about $10 to $100 per dose. If you extrapolate this to what this might mean for creating new opportunities for making medicines when and where we need them, it's instructive to look at the numbers. So we believe that this IVAX platform can present a portable, inexpensive, and rapid biomanufacturing approach without a cold chain. And we think that because of the following. If you imagine that a dose of a vaccine is about 5 micrograms of antigen, and I mentioned a slide ago, in the paper we just published, we were producing about 0.1 grams antigen, 0.02, so 20 micrograms per milliliter, grams of antigen per liter. Recently in work I'm not showing you, Katie Werfel and Jazzy Hirschway have actually increased these protein expression yields to 0.1 grams antigen per liter. So this is a manuscript we just had accepted at Nature Communications. And we made this in an hour, right? So if we think about how fast can we make vaccines, certainly in pandemic response, this can be important. If we make 0.1 grams of antigen per liter per batch, imagine we could make 2 batches per day, because you could do 1 in the morning and 1 in the afternoon if they're each only an hour. And a dose is 5 micrograms, or 5 times 10 to the negative 6 grams. That means in a single liter, you can make 40,000 doses of vaccine. If you scaled that to 1,000 liters, so a meter cubed, right? Think of that kind of a space. I mean, we can buy large bags of compost if you really go in some places for that size. You can make 40 million doses per day. If you extend this using that same 1,000-liter bioreactor for roughly 3 weeks, 3.5 weeks, you can actually make enough vaccine for 1 billion people. So for 9,000 liters, we can actually imagine in about 3 weeks of time, making enough doses of a vaccine against a pathogenic bacteria in this glycoconjugate approach for the entire planet. And the cost at the moment is about 50 cents per dose with some of the innovations that Katie has brought to the group. And that compares favorably to a recent vaccine rollout of the Menafrivac vaccine, so for meningitis, that worked through Africa in 2010 to 2016. So we're really excited about where this process is going. We imagine how can we best share resources, enable local production, perhaps even create new models for open reagents to move out into the world different opportunities for biomanufacturing of medicines and vaccines. So rethinking ways to distribute global medicines across the globe is clearly important, but there are really many global challenges to think about addressing. Obviously, at the front of all of our minds is COVID-19, and I will tell you my laboratory also has efforts in COVID-19, both in diagnostics and manufacturing and discovering neutralizing antivirals. But what I'd actually like to spend kind of the second part of my talk on today is focusing about water. Clean water is an increasingly precious resource on our rapidly warming planet, right? More than 2 billion people lack access to clean water today. And by 2030, it's expected that about 50% of the planet will live in an area where there's water scarcity. Target 6.1 of the United Nations Sustainability Development Goals has as its goal to achieve universal and equitable access to affordable drinking water for all. And if we're going to reach this goal, one starts to wonder, especially in times of water scarcity, shouldn't everyone be able to know what's in their water? And so in some collaborative efforts with Julius Lux, we've been trying to think about new diagnostic strategies for water contaminants. And existing strategies really put testing out of reach of people that need it. They can be inexpensive, inaccessible, and slow. They can be qualitative or not robust. And we'd like to imagine, and in particular, innovations that were led by Adam Silverman in the group, if we could create inexpensive, rapid, and quantitative technologies that let everyone know what's in their water. Now, when you look to nature, of course, has developed numerous strategies to sense and process information. Many types of biosensors exist in biology. Of course, whether we are teaching our immune system as a bacteria about invading pathogens, or you're a macrophage here chasing this bacteria in this very classic example of a movie. Unfortunately, nature's solutions for biosensing are oftentimes locked within microorganisms. And so our solution is to think about these freeze-dried, cell-free diagnostics. So in a sense, leveraging the same types of cell-free freeze-dried systems I mentioned a minute ago that we were producing for vaccines, but now transitioning these to freeze-drying genetic programs that can help us detect small molecules, rehydrating those sensors with a test sample and knowing within an hour whether or not you have, let's say, a small molecule chemical contaminant. And I'm showing an image here of a picture from some work that Julius Lux published in Nature Biotechnology with Khaled Alam and Kirsten Young from his lab, focused on developing different types of biosensors. And these cell-free sensors are actually quite exciting. They're sterile, they're non-GMO, they're programmable, lightweight. They have low to no power requirements. They can be shelf-stable and inexpensive, pennies per reaction. So over the course of the last few years, Adam has really been developing ways for enzymatic outputs and green fluorescence to enable the detection of a variety of chemical contaminants in water. And so I'm just showing here the development of a variety of sensors for 6 of 10 chemicals of major public health concern that have been identified, from arsenic to atrazine to chromate, copper, lead, and even nitrate. So we think that there's really an exciting innovation space to imagine using cell-free systems for diagnostics, in particular in water quality control. Now, these cell-free diagnostics offer a shift in how healthcare systems might be able to adapt to public health emergencies, certainly might help us understand challenges surrounding water scarcity in the future. And they could offer low-cost healthcare globally for identifying other types of pathogens or disease. Interestingly enough, they also lead to new opportunities for educational literacy and citizenship. And so I'm extremely proud of an effort by many people in my lab, and I know I didn't fit everyone on the slide here. We're really focusing on trying to create new models for innovation in education. Scientific literacy is, of course, critical to our discipline. Certainly, we all believe that everyone needs to be able to read and write. I think, we're all behind. We need to read and write in computer code. And I think it becomes increasingly critical that we can all read and write in DNA. And so how do we enable a community of scholars to grow up in the system and be inspired by STEM education opportunities? And so Adam and others have been developing these same types of sensor systems to inspire and work with students, even in at-home testing kits we're exploring at the moment. Now, this got started because hands-on science activities improve student engagement and understanding. We know this, right? Whether it's classic chemistry kits or my kids have played with snap circuits. And one of the challenges with synthetic biology is that biology oftentimes requires specialized facilities and equipment. It can be prohibitively expensive. Do you have sterile technique or not? And few topics in biology reflect current interests like antibiotic resistance or CRISPR. And so in a collaborative effort with Jim Collins, led initially by Jessica Stark in my lab and Ellie Huang and Peter Nguyen in Jim's lab, we developed these BioBits kits, which are user-friendly, just add water approaches to teaching synthetic biology. They can be about $100 per classroom kit for 30 students, include key lab equipment, teach cutting edge topics, and already they've been spun out to reaching more than 10,000 children. This is just an example of what some of these systems can look like. This is just add water design and execution of in vitro genetic programs by untrained operators. You can kind of think of this like, the biological equivalent of a light bright kit, where we provide students freeze-dried cell-free reactions, plasmids encoding different colors, and allow them to create and paint the systems that they wish, right? And they've come up with, pictures like a rainbow. This is a periodic table. My 6- and 8-year-old sons at the time played a game of Connect 4. You can see the red team won down here, and even tried to create with some of our local collaborators in Evanston, the Evanston High School Township Wild Kit. So I think that building STEM education literacy becomes increasingly important, helping students engage, pursue careers in this direction. And it's really critical that we as a community facilitate opportunities and outreach for synthetic biology, right? Many of our leaders in Congress and others, were past schooling by the time recombinant DNA technology really came to bear. And so making sure that we really provide educational opportunities to inspire others to pursue STEM is important. So I think as we think about things like distributed manufacturing of medicines, on-demand sensing for water quality control, and even inspiring others to get into education, we of course need to ask ourselves, what other problems might we wish to solve? And I guess that's where I'd like to leave it for today. I was asked to present for about 15 to 20 minutes, and I think that there's just a lot of exciting opportunities to move beyond cells to try to imagine how we create a fair and sustainable 21st biocentury. And I would challenge us as a community to think about how we build things like human capacity to support biological science, engineering, but also policy, security, culture, ethics, art, and leadership needs of the community. How do we build tools and knowledge to make biology easier to engineer and accessible to all? How do we develop and build applied technologies across scales that solve some of those problems I mentioned at the very beginning in climate change or ensuring that medicines are accessible to all? And how do we imagine creating a world in which we all wish to live? I think that these are important questions that we and others really must think through as we imagine how our technologies can shape the future. So I'm very grateful to my group for providing a culture of innovation and creative place to work. This is a little bit of an older picture. We're still waiting, like many of us, to get one of those new group pictures where we all get to stand next to each other. And very grateful also for the support by many funding agencies that have allowed us the freedom to explore the types of research that I mentioned. I also just want to highlight, the growing presence of the Center for Synthetic Biology on campus. We cross, for example, the School of Engineering, the School of Arts and Sciences, as well as the medical school and many, many departments at this stage and are growing. It's really a wonderful community of scholars and encourage you to connect to them as you see fit and the opportunities that come by. Also very grateful for the Chemistry of Life Processes Institute and the environment in which it has provided and created for all of us to pursue science and push the boundaries of where the future might lie. So happy to stop there, take questions, and look forward to a discussion.
So Mike, would you like me to read off some of the questions in the chat? This is Lisa, or would you just go ahead and like to do that yourself?
I can easily churn through the chat. So a few questions that have popped up. So for the conjugate vaccines, are we purifying them before we inject them? Yes. So at the moment, we're actually purifying the conjugate vaccines in the mouse study prior to injection. Though interestingly enough, the E. coli strains that are glycocompetent that we have engineered actually have low enough endotoxin activities that they, in theory, could be directly injected, though this would be a new model of a therapeutic and would have to be studied in great detail to ensure safety. In terms of the cell-free system for synthesizing lipids or proteins that are water-soluble but found in membranes, we indeed can produce many proteins that are membrane-bound in the system. In fact, the glycosylation system only works because of membrane-dependent biology. So it turns out that the periplasmic wall has these 13 transmembrane domain glycosyl transferases that are embedded in them and active. We also activate oxidative phosphorylation in the system to fuel energy regeneration. Another question in the chat is, at what cost rate and how can businesses thrive, especially if we try to move price points down? This was just a private question to me. I think that I'd actually love to get to a point where we could make conjugate vaccines or other vaccines using these cell-free systems for less than a penny a dose. And I just would love to see how and in what way that impacted the models. Now, Katie already in the last year or so has been able to drive innovation down in costs by more than an order of magnitude. And I think even potentially within her PhD, we may be able to get down to, 5 cents a dose. We'll see how far we can push things. But I'm excited about where that is, and I'd love to challenge the model and see how we can open access to medicines. Another question is about, it seems like 3 different temperatures provided exactly equivalent activity. Do you have a preferred temperature for using the IVAC system? Right now, I think in lab, I actually don't know. We probably run it at 37 degrees, but you're right, at least on the short time scales, it doesn't really matter so much. We're making about the same amount of protein, and that's important. In terms of a question, fascinated by synthetic biology, what are the best ways to dive into synthetic biology? So there are lots of entry points. Certainly the Center for Synthetic Biology on campus has some student groups, in particular gene mods, and I would encourage you to explore that. More broadly, the Engineering Biology Research Consortium, which I help co-lead as the academic co-lead, provides kind of a resource for many synthetic biology academics. And if you're looking more on the industrial side, I could encourage you to take a peek at something called SynBio Beta. And that will give you a landscape of the emerging industry around synthetic biology and how that's growing. Thinking beyond 10 years, do you see cell-free systems as occupying a niche role for portable high-value production or a larger scale centralized production? I'll be honest, I think the jury's probably out on how and in what ways cell-free systems will manufacture both medicines, but also biochemicals and other products. From a medical perspective, there has still yet to be an FDA-approved product that's come out of cell-free systems. There's a company called Sutra Biopharma who's moving forward an antibody drug conjugate. And I think right now the data look promising. And if that happens, I think it will kind of open the floodgates towards opportunities in making medicines by this approach. And that's really kind of needed for the field for it to expand. I also think that there are exciting opportunities for synthesis of sustainable chemicals or carbon negative chemicals to address climate change using cell-free systems. Interestingly enough, we find that we get much higher volumetric productivities than gram per liters per hour of small molecule chemicals in this space. But I don't see it replacing what exists in the industry right now. But I do see it adding as a complement to new directions and new manufacturing approaches that might be there. In terms of purification strategies for on-demand or portable applications, there are purification strategies you could imagine using inexpensive things like carbohydrate binding domains attached to cellulose. And in fact, Govind Rao, who's at the University of Maryland, Baltimore County, is advancing a vision for on-demand synthesis of medicines in lower resource areas where even parts of the purification components are built from sustainable and locally sourced products. And so I think that there are some opportunities there, and certainly many dimensions of the problem that can be focused on. In terms of Northwestern being positioned in the nation and the world in this field, this is a question that just kind of came towards me. How is it, in this field, and what's missing to support next generation synthetic biology? I would argue that if we're taking the field as synthetic biology, Northwestern at the moment is positioned quite well. We have efforts that cross both the emerging areas in cell-free synthesis and diagnostics, a variety of AI, machine learning guided approaches, protein design to modulating manufacture of molecules. A variety of colleagues are looking at things like bioplastics degradation, which is becoming urgently important. And we also have quite a few folks looking at design-driven medicine. So I think as a community of scholars, we cross everything from sustainability to design-driven medicine and these features to diagnostics. So it's really an exciting time, and I think we're positioned well. In terms of there are growth opportunities, and in the end, I think one of the things we all love about Northwestern is the people. So I think that continuing to bring in great people will help drive exciting futures for us here at Northwestern and in the field. In terms of can the technology be applied against viral infection, you could imagine synthesizing spike proteins as antigens, right? Or you could imagine expressing antigens for Zika or other components like this with this platform. They wouldn't have to be a conjugate vaccine. But I think we like conjugate vaccines because there is this special feature of the sugar. Additionally, I mean, the emergence of RNA vaccines, I think, is going to transform how we think about vaccination. And the amount of data we're getting on safety and efficacy profiles by trying to vaccinate populations of countries in the current model with the RNA vaccines is certainly going to put things in the future. Though I would say, I come back to the need for manufacturing capacity as being important. I think that when people look back in history, this moment for synthetic biology and biomanufacturing will have been a really critical one, because synthetic biology and biomanufacturing are delivering on the production of medicines at a global scale at a rate that's never been seen. I mean, last week I was talking to my biochemical engineering class about, okay, so it takes, multiple $1 billion in 15 years to make a medicine. And this is the canonical historical view in a sense. And that model has been disrupted in this current pandemic. And the implications of that, I think, are still somewhat unclear. But it provides an exciting, hopefully, future where we might be able to help folks certainly across the globe. Okay, I think I'm to the last question. If I missed one, Sheila or Lisa, you'll have to correct me or point me to it. Can you think of difficult synthesis that may use cell-free systems from 2 or more organisms? This is a super interesting question. Indeed, one of the really kind of fun parts about cell-free systems is you're not limited to one organism. So you can kind of create organismal amalgams of extracts and perhaps get the best of multiple worlds. And we and others have done this, for example, looking at the expression of hard to express proteins, let's say P450s in eukaryotic organisms, with bacterial organisms providing other comparative elements. So I think that there are exciting opportunities to kind of merge the best worlds for either discovery, which honestly, I think when you look at cell-free systems historically, they've been looked at as a discovery platform. And our lab has many discovery platform focal points as well. How do we identify the best sets of enzymes for glycosylation? Or how do we identify the best antiviral against COVID-19? Or how do we identify the best sets of enzymes for production of a sustainable chemical? And many basic science questions, but there is this emerging appetite and opportunity, I think, in manufacturing. Okay, so one last question coming in here, and then happy to address others, but how do you think about the effectiveness of the vaccine using synthetic biology compared to traditional manufacturing? from being cost effective in large scale production. I think what's interesting, in the end, the effectiveness of these RNA vaccines at greater than 90% is basically best in class, right? I mean, we are at the level of where the best vaccines have been. In terms of cost effectiveness and scale, I probably haven't analyzed it well enough yet, Vinay, to have a good answer for that question. And is there an upper limit of size for proteins you've made with cell-free protein synthesis systems? I think the largest protein we've made is on the order of 400 kilodaltons. So that's pretty big. These are NRPS, PKS type systems. Those are harder to express, just like they would be in cells. And honestly, protein expression is still somewhat of a black magic art. But cell-free systems, we think, provide some advantages for molecular control that we certainly are excited about. Okay. Other questions? Happy to address these. What's the limit in terms of replacing industrial feedstocks for plastics? This is a good question. I think that in terms of feedstocks, one of the challenges to think about, and we are working on with collaborators, is how do we move towards, not necessarily sugar-based feedstocks, but things like carbon dioxide, carbon monoxide, sunlight, et cetera. And I think there's a shift thinking in this direction, and capacity is growing. So we'll see where it goes in the context of manufacturing products in a domestic setting. Lonzatec, certainly, if you're interested in learning about CO2, CO conversion from syngas, have some exciting developments recently, and you can look that up if you're curious. Well, this was fun. I don't know that I've ever answered that many questions in a chat without seeing anyone's faces. So I'm really sad we didn't get to do this in person. And hopefully next time we can have a little more personal interaction, but otherwise grateful for everyone's time. Hopefully this was interesting. Thanks again to my group for all of their amazing contributions to what we do. And of course to Sheila and Lisa for having us here today. Very grateful to have the chance to share.
Thank you, Mike, for a terrific talk, and everyone for your participation. Please check out more news about Mike and his lab at the CLP website at clp.northwestern.edu, and sign up for our newsletter or follow us on Twitter, Instagram, LinkedIn, or Facebook. Mike, thanks again. It was just wonderful.
Thanks, everybody.
Bye, everyone. Take care.
Thanks, Mike.
Be well.